Cardiovascular disease, specifically myocardial infarction and ischemic stroke, remains the leading cause of mortality worldwide. Many of these acute events are driven by impaired fibrinolysis. When the body loses its ability to dissolve blood clots efficiently, a minor blockage can escalate into a lethal thrombotic occlusion. The specific molecular culprit behind this failure is Plasminogen Activator Inhibitor-1, or PAI-1. It acts as a rapid-fire inhibitor, neutralizing the enzymes that would normally break down fibrin networks. Because elevated PAI-1 is tightly linked to thrombosis, geneticists isolated a highly studied regulatory variant in the promoter region of the gene, controlling its production, known as the 4G/5G polymorphism. To understand a patient's risk, we have to look past the macroscopic clot and examine the microscopic regulatory elements buried within their DNA. In 1995, molecular biologists established a mechanical proof. Working in controlled cell cultures, they demonstrated exactly how the 4G version of this allele increases the baseline production of PAI-1. Yet, when epidemiologists tested this mechanism against massive human cohorts a decade later, the data fractured. Different populations yielded highly contradictory stroke risk profiles for the exact same genetic sequence. Today, we will unpack the molecular mechanics of the PAI-1 polymorphism, analyze the conflicting population data, and examine why a flawless in vitro mechanism does not always translate into a reliable clinical predictor. This tension between the petri dish and the population forces us to reconsider the effectiveness of using single-gene models to predict complex cardiovascular risk. The molecular foundation of this story begins with a 1995 study published in PNAS by Erickson and colleagues. They sought to determine how PAI-1 transcription is regulated at a basal level. They located the variation exactly 675 base pairs upstream from the transcription start site of the PAI-1 gene. Looking at this sequence alignment, we can see the physical nature of the mutation. The 5G allele contains a 5-guanosine tract. The 4G allele is a single base pair deletion, resulting in only 4 guanosines. This microscopic structural variance, the absence of one single nucleotide, forms the physical basis for the entire cardiovascular risk hypothesis. To determine if this missing guanosine had functional consequences, the researchers used an electrophoretic mobility shift assay, or EMSA. This technique separates protein DNA complexes based on their size and charge. This gel captures the interactions. The dark band A indicates both alleles bind a thymine transcription factor, but there is a critical anomaly lower down. An additional protein complex, factor B, binds exclusively to the 5G sequence. In the 4G lane, that space is completely empty. Because the 4G allele correlates with higher levels of PAI-1 in the blood, the researchers hypothesized that this 5G-specific protein, factor B, acts as a transcriptional repressor. The single missing guanosine in the 4G allele completely abolishes the binding affinity for this secondary regulatory protein. Using footprinting and methylation interference assays, the researchers mapped the exact physical boundaries where factors A and B attach to the DNA. What they found was a mechanism of steric hindrance. Factor A, the activator, and factor B, the repressor, require overlapping binding sites on the promoter. The 5G sequence has enough physical space to accommodate both proteins simultaneously. This allows factor B to physically interfere with the activator, restricting insufficiency. To measure the actual transcriptional output of these two competing models in a living cell, they used a chloramphenicol acetyltransferase, or CAT, reporter gene assay. This table quantifies the results. Without the repressor to interfere, the 4G construct yielded a fold induction of 33.7, more than double the 15.3 induction seen in the 5G construct. The molecular model was finally complete. 4G homozygosity definitively increases basal PAI-1 transcription in a tightly controlled cellular environment. If the 4G allele produces more PAI-1, it should predictably increase stroke risk. The next step was observing how this verified transcriptional increase translated into clinical outcomes in human populations. A major test of this hypothesis came from nested case control studies conducted in northern Sweden, which tracked healthy individuals to see who eventually experienced a first-ever stroke. The Swedish data aligned perfectly with the molecular theory. Individuals carrying 2 copies of the 4G allele demonstrated a significantly increased risk of ischemic stroke, yielding an odds ratio of 1.87. However, a separate study analyzed the exact same polymorphism in a Dutch cohort. Their findings showed an absolute null association. The Dutch data returned an odds ratio of exactly 1.0, meaning the 4G genotype conferred 0 additional risk for an ischemic stroke. The cleanly proven molecular mechanism from the Petri dish failed to hold absolute predictive power across distinct human populations. To reconcile this, researchers looked deeper into the Swedish cohort data to find variables that might be modulating the genotype's actual penetrance. They found a critical modifying variable, the systemic level of triglycerides in a patient's blood. This revealed a strong gene-environment interaction. The risk did not rise linearly with genetics alone. Instead, individuals with both the 4G homozygosity and hypertriglyceridemia experienced an exponentially compounded risk of stroke. The 4G allele creates the biological capacity for higher PAI-1 transcription, but that capacity requires specific metabolic stressors, like high triglycerides, to be realized as a clinical event. This paradox reoriented how researchers approach cardiovascular genetics. Disease etiology cannot be reduced to single gene determinism. Because environmental interactions heavily dictate the final clinical outcome, modern pharmacology has largely shifted its focus away from targeting isolated promoter variants. Therapeutics have evolved to modulate the broader metabolic environment. Current drug development is heavily focused on managing severe hypertriglyceridemia and clearing remnant particles from the bloodstream using emerging RNA therapies. The path from a 1995 footprinting assay to the large-scale trials of the 2000s maps the distance between a localized molecular mechanism and its systemic expression in a human patient. A flawless molecular proof is only the first step. The real challenge of medical genetics lies in mapping how pristine mechanical pathways survive the messy, interconnected noise of systemic human biology.